Good morning. Can anyone hear me? Yay. It gave me that same warning. Maybe I gotta update Zoom. It's very frustrating because it's like you show up, you're like, ready, okay, I'm gonna teach this class. And then it's like, warning, nobody can hear you. It's like, okay, thanks, Zoom. Thanks for that vote of confidence, Zoom. Okay, we gotta get through a lot of things today. We gotta catch up for a little lost time so that you guys are well prepared for Monday's lab. Let me pull up everything we need to pull up. Okay, so we're going to share my screen and we'll go to liverworts, hornworts. We're going to finish that up first. Okay, good. Awesome. Before I do get started, does anyone have any questions at all about anything? We had a big blizzard here yesterday. I don't know if you could see the winter wonderland that's outside. All right, let's see, pull this over here. I think I have a question here in the chat box. Yes, if we attend lecture, do we still need to post in the discussions? Yes, we do need to do that. So there's two things. There's attendance and there's participation. Last class, somebody asked if you participate in the live lectures, do you still need to post in the discussion boards? It's easier for me to keep track if you do. And as I mentioned last time, if you participate in class, and let's say you asked a question during class, you could even write in the discussion board like, oh yeah, I really liked the question that I asked in class, just to remind me that happened. Because I don't save the chats, which I could, I guess, fix that, but it's also good. Discussion board's good because it also lets your fellow students, so everyone can see everybody's posts, and everybody could actually write on everybody's posts as well. So the discussion board thread is cool. Yeah, like Hawa did, exactly. Hawa did great. She participated in class, asking questions, and then she went on the discussion board and wrote, man, I loved all the questions that I asked today. And that's perfect. Do that. Yes. But it's also cool on the discussion boards also because a lot of times students have the same questions. My experience is that the same things confuse students. And so it's good to, it's a place that your fellow students can see how everybody else is dealing with the material, and you guys could even discuss things if you want to back and forth there. All right, we gotta go. We gotta get going. So if no one has any other questions, well, if you have a question, feel free to throw it in the chat. All right, let me make this. So we're not going to go through this again. So last class, we're not going to go through this again in great detail. But last class, we covered the liverworts, you might remember, right? Phylum Marcantiophyta. And we kind of finished class by going through this alternation of generations in the liverworts, right? Right, spores grow into the gametophyte plants. Gametophyte plants make sex organs that make egg and sperm, and then egg and sperm come together to make the new sporophyte plant, right? This is what we went through last time. Sporophyte plant grows from where the egg was. So sporophyte plant grows from the archegonium, the gametophyte plant. There it is there, hanging upside down like a bat. This is it close up. And then the last time, right, we were kind of just starting to talk about these things called elators, which are these kind of springs that allow the capsule or the sporangium to open better so the spores can leave. Sorry, I didn't have any coffee before class, so I'm definitely going to be drinking coffee. It's a cross section through a capsule or sporangium showing you the spores that are there waiting to be released. Remember in a previous class, I mentioned this word tetrad, which is just a fancy word for four. So after, let me just do this real quick. Remember, just so you remember, remember when you have the spore mother cells that are 2N and they undergo meiosis and make haploid spores. But when you go through meiosis, as we learned in the, I think, second class, so you can go back to there for a refresher. When you go through meiosis, you end up with 4 things at the end of meiosis, 4 daughter cells. And so hence, these spores are the result of meiosis. And so each of them initially will be in a group of 4, which we call a tetrad. When you look at a tetrad of spores under the microscope slide, it always looks kind of like a circle with like a peace sign in it, like that. That's a tetrad. The reason why is because if you have the 4 spores kind of like stack on top of each other, I don't know if you've ever played like pool and like tried to stack the balls on top of each other just for fun. But like if you were to take 1 spore here, 1 spore here, 1 spore here, there's 3 spores. And the 4th spore will be stacked on top. And anyways, because of that configuration, when you look at the tetrad of spores under the microscope, it looks like that, because you're basically seeing this here. I'm drawing it in black, give or take, so something like that. But that's what that little crease is in those tetrads. Anyways, spores inside of a sporangium. These are those elators again. So these snaky things are showing you. So here's a elator that's wet. Once the capsule opens and exposes the contents of the capsule or sporangium to air, that starts to dry out. And as that starts to dry out, the elators will start to dry out. And when they start to dry out, they kind of start to coil around. You like that? That's my elator movement. They start to coil around. And when they do that coiling and movement, it just kind of helps the spores jump out of the sporangium. Yeah, okay. With this, we saw last time as well. This is just a real microscope slide showing you spores and elators. The long spring-like things are the elators. The purple and green things are spores, probably at different stages of development, meaning some of those spores are probably ready, some of those spores are not quite ready yet. All right, we also went through this last time. This was just a nice side-by-side comparison of the sporophyte plants in the true mosses, which is on the right here, and the sporophyte plant of the liverwort. Main take-home message of this slide was that the components are the same in that you have a foot, a seta, and a capsule for the true mosses and the liverworts. Main difference, though, is that the liverwort sporophyte is hanging upside down from the arms of the archegoniaphore. And so it hangs upside down like a bat. All right, so that's, so far in this class, we've only talked about sexual reproduction. For example, the egg and sperm coming together and fusing to make a 2N zygote. Sexual reproduction is important because it allows you to recombine genetic material and maintain genetic diversity. So that's why we do it too, right? It's important. But you don't, in plants, you can reproduce or make, in other words, reproduce meaning make new individuals. You can reproduce it in plants a lot of times asexually, So the prefix A, if you ever see A in front of a word, means without. Without. So the word asexual reproduction means reproduction without sex. So no egg, no sperm, no gametes, right? And a lot of plants are able to do asexual reproduction. And the result of asexual reproduction is always going to be clones. Always going to be clones, because you're not combining egg and sperm. So you're not combining genetic information from one individual with genetic information from another individual to make a new individual. You're simply cloning one individual using asexual reproduction. And there's different types of ways, right? So this is what I just said. No egg and sperm are not involved. And you get new clones, new plants that are clones. So keeping that in mind, I'm going to ask the class now here. So hello, wake up, put down TikTok for 1 second. I'm going to ask you a question. So if we have asexual reproduction, which is reproduction that does not involve any egg, does not involve sperm, right? What process must be involved in order to have asexual reproduction? So far we've learned about 3 processes in this class. We learned about meiosis, we've learned about mitosis, and we learned about fertilization. So of those 3 processes, which process is involved in asexual reproduction? Yay. And a lot of people in the chat box, thank you guys, are saying mitosis. Correct, because absolutely, yay, across the board, because no egg and sperm's involved, so we know it can't have anything to do with fertilization. And you're making a new individual from an existing individual. And so what that means is you're basically taking a piece of material, and that material is going to grow into a new individual, right? And so you guys all got it right. It is mitosis. Thank you. And cytokinesis. Yeah, but very good. Okay, so there's different types of sexual reproduction. Today we're going to talk about the types that are in liverworts real quick. So in liverworts, there's a couple different types. There's fragmentation of the gametophyte plant body. So fragmentation just means the gametophyte plant gets ripped apart. So if you could think of like your gametophyte plant, you're living near a little river, and like, I don't know, something comes along and like steps on you, right? Maybe a little deer is like heading down to the river to like eat or drink water or something, and its little hooves like smash all the gametophytes apart. When that happens, each of those little pieces of gametophyte plant can actually grow into a new individual gametophyte plant via asexual reproduction. So like each little fragment grows, right, mitosis, into a new plant individual. So that's super cool, because it's like basically if you could like rip off your finger and like throw it across the room, and then like over there is a new me, like growing from a new clone of me from my finger. Thank you. Hi, you know, that's super cool. So this fragmentation of the gametophyte plant body, for what it's worth, liverworts do this, but hornworts and mosses can also do this fragmentation of gametophyte plant body thing. If you rip up a moss, a lot of times you can do this can occur as well. But liverworts in particular have a special structure that's called gemmacup. And inside of the gemmacup is gemmy. And you say it gem like truly outrageous gem, right? So gemma cup and gemmy is how you would say those words. A gemmacup is basically a cup, a little cup-like structure that's on the body of a liverwort gametophyte plant. And the gemmy are kind of like these little jelly bean looking things that are inside the cup. And they will will come out of the cup and be able to make new liverwort gametophyte plants. And some mosses do have this as well. But we mostly only see them on the liverworts in this class because of the mosses we cover. So here they are. My head may be covering some of the word. My head is covering the word gemmy, just so you know. So here, each of these, let's see, what's a good color? Maybe, I think black. Black is good. So this is a close-up of a liverwort gametophyte plant, right? This is a thalloid-shaped gametophyte plant. It's flat and green. And you see all these little cups, right? So like here's a cup up here. Here's a little cup here. Those are the those are the gemma cups. And inside of the gemma cups, the little green things inside are the gemmy. So I think I have, this is a microscope slide through a gemma cup and containing gemmy. So here is the cup part. And then the little green jelly bean looking things are these inside here. And each of these is a gemmy. So what happens is, once again, thank you, rain. Rain or some other source of water will fly into the gemma cup, right? Splash into the gemma cup like this. And when that happens and it splashes back out, the gemmy will also like fly out. And then when they land somewhere, then via the processes of mitosis and cytokinesis, they can start to divide and ultimately make a new gametophyte plant via asexual reproduction. So I think I have a little video here. I do. Let me grab that. And I'm sure there's going to be an ad because it's such a popular topic that people watch. Yep. Okay. No ad. Don't listen to the ad. Okay. Skip ad. Okay. Now hold on. I know I haven't shared it yet. 1 second. Okay. So I'm going to stop sharing that, and I will share instead here we go. Share this. Okay. Make it big. All right. So you guys should be able to see this video. Let me know if you cannot. And so anyways, this right here, this is one gemma. This is one gemma cup. And inside, I don't know if you could see, so this whole thing here is the gemma cup. And inside here, you see these little like darker spots, those are the gemmy. And so what this person is going to do now, they're going to drop a drop of water into the gemma cup and boom, there you go. See, and the gemmy fly away. We watch it again, boom. And so each of those gemmy are going to fly away and grow into a new gametophyte plant individual that will be genetically identical to whichever plant individual this is, right? Because this is via asexual reproduction, so there's no egg and sperm involved. And you'll probably see, so if you look at the diagrams we've looked at so far for liverworts, a lot of times they show you the gemmy cups. And here, share this. So for example, here is a good one right here. All right, you should be able to see the PowerPoint slides again. But so for example, here, this is our cartoons again, right, of moss, liverwort, and hornwort. And just so now that you know what they are, look, gemma cup, right? There's another gemma cup, and here's 3 gemma cups. So if you look back at all the other diagrams that we've seen so far of the liverworts, you'll notice these little cup-like structures. That's what those are. Okay, that's asexual reproduction. Now we got to move on to the hornwort. Finally, so you'll see, you saw moss in lab last time, right? Moss and peat moss. In lab this coming Monday, you're going to see liverworts and hornworts. So let's get through those hornworts so you can be prepared. So hornwort. Liverwort was liverwort because somebody whoever described liverwort thought that the gametophyte plants look like a liver, right? Well, whoever described hornworts thought that the sporophyte plants resembled horns. And so the word hornwort is basically a horn plant. And let's see, so just so we got it here, down here, this is the gametophyte of the hornwort. And then these horn-like structures coming up are the sporophyte plants. Oops. So a plant with horns, right? Clear all drawings. Yes. Here's what they look like on a rock. So just so you guys know how to pronounce this word. So the phylum for hornworts is Anthocerophyta. Anthocerophyta. One way I try to kind of remember it myself is that, I don't know if you guys have ever, if you know about dinosaurs and you've heard of a triceratops, right? It's actually ceratops, but triceratops. What does a triceratops have, right? Tri, 3, and ceratops, 3 horn structures, right? And so that helps me remember this Anthocerophyta because it's a horn, right? So triceratops has 3 horns, and Anthocerophyta is the liverwort phylum. That's how I remember that. But yeah, if you look at this rock, all the green flat stuff is hornworts and the hornwort gametophyte plants, and then these horn-like structures are the sporophyte plants coming out. So you may notice already that the hornwort gametophyte plant is also flat and green. It's also flat and green like the liverwort gametophyte plant, the phalloid-shaped ones. All right. Yep, this is what I was saying. So that the etymology of that word cero refers to horn, so that's why it's called that. Antho means a flower. Don't worry about that yet, but we will encounter that term again. But it's basically a plant that sprouts or flowers horns. But there's no flowers involved here. Super old plant. You're looking at close to 400 million years ago. Based on the fossil records, or probably even earlier than that. There's not that many species of hornwort. There's only 300-ish that are known. Because it's a non-vascular plant, we know the gametophyte plant is dominant. And the most common species that you will encounter is Anthoceros, which is what you will see in the lab as well. And it's nice. It's nice, like remember Marcantiophyta, we had Marcantia, and then now we're in Anthocerophyta and we have Anthoceros, right? So that's nice. It's like you don't have to memorize double things. It's either nice or confusing depending on how you want to feel about it. Similar to the mosses and the liverworts, hornworts, they need water to do asexual. They need water to do for the sperm to swim. Hornworts, they need water for the sperm to swim, just like mosses and liverworts. So they tend to live in these tropic and temperate environments. And they are also early successional, which we've learned about in previous, meaning that they can be some of the very first plants to show up in new habitat. Just like the mosses and the liverworts we looked at, some hornworts are epiphytes, meaning that they can grow on other plants, usually trees or shrubs, right? Usually trees or shrubs. This here is this whole thing here. Whoa, okay. Okay, thank you. This whole thing here is a hornwort plant, both plants. You got the gametophyte plant and the sporophyte plant. Maybe white might be better to use here. Gametophyte plants are the green flat things. On the bottom, sporophyte plant horns are emerging, right? Here's one horn. Oops, here's another horn. Here's another horn. Some of these sporophyte plants are already, the capsule's already starting to open. Like, so here, for example, you see this is the sporophyte plant horn coming up, and the top of the horn is like brown and twisted. That's what it looks like when it's starting to open. We'll see that on subsequent slides. But just like liverworts, you can find hornworts in urban environments too, like Newark. So I forgot to mention last time when I mentioned the liverwort, seeing liverworts in Newark. When I saw liverworts in Newark, that when I showed you last class, that was my first time ever seeing liverworts in the wild, as it were. I had seen them at botanical gardens. I had seen them in lab, things like that. But I had never been in my life before that moment walking around and then, oh, what do you know, liverwort. So my very first time seeing liverworts was in Newark, New Jersey. And likewise, my very first time ever seeing hornworts in the wild was also in Newark, New Jersey. So here on the left, it's hard to see, but I actually had this was near Smith Hall, like near the steps. And at the time I had a foot injury, so I was on a crutch and I couldn't climb over the little step area to go take a picture. So this was as close as I could get. But all these little like pokies coming up, those are hornwort sporophyte plants that were just starting to emerge. But yeah, so look down when you're walking around Newark, especially in the spring when it's really wet and stuff, because you will see these little guys around if you know where to look. Okay, Anthocerophyta, hornworts. Here is a nice picture of both the Gametophyte plant and sporophyte plant. So this here is probably like, this is probably like 6 or so Gametophyte plants, 2, 3, you know, there's several Gametophyte plants down there, but and then these horn-like structures are the sporophyte plants. First, let's talk about the Gametophyte plant. I was like, now this class is making me look closely at plants everywhere, anywhere I go. You know what? I'm telling you, this is a phenomenon that happens with this class. Paula says, right? You guys aren't alone. This is why I've actually fought to keep this class, because, you know, especially if you grew up in a very urban area and you may have just been walking your whole life, never even noticing the plants around you. And so I often get feedback from students that now they realize, like, whoa, I'm surrounded by plants, which is just part of the reason I keep this class going. And I've really made the department not lose this class, because that's an important experience. I think if you're living your day-to-day life and you can have something occur like a class where you're like seeing the world, seeing new organisms in the world that you never saw before, and that's pretty cool. All right, so can you show the Gametophyte or the, yeah, so we're gonna see these on many slides coming up. Kiri, is it Kiri? I think Kiri. Oh, Kiro. Dang, Kiro. Sorry about that. Yeah, so we'll see these on a bunch of slides coming up, but yeah, this one here is nice because you can see, like, so the flat green part is the Gametophyte plant. This is actually probably several Gametophyte plants, and then the horn-like structures coming up are the sporophyte plants. But we're gonna see lots of these coming up on the next slides. All right, first, and here we go. Here you go. We're gonna see some more right now. First, let's talk about the Gametophyte generation. So Gametophyte generation, we're in the non-vascular plants. So we know the Gametophyte generation has to be dominant. So the main thing that you see, and because also non-vascular plants, you know, there's no true roots, no true stems, no true leaves, and there's no vascular tissue, right? Right. When we looked at the mosses and the liverworts, we talked about how the mosses from using the naked eye are a little bit more complex than the liverworts. And the liverworts, using a microscope, have more complex cellular structure. But in terms of the hornworts, the gametophyte plant, it's really simple. I mean, it's basically a green blob. When you see the preserved specimens in the lab, you'll get an appreciation for this. It's literally like a little green blob. So there's no complexity going on really in the hornworts in terms of the gametophyte plant. Yeah, it's basically a little green blob. So it's photosynthetic blob and has one large chloroplast in each cell. Very simple plant, gametophyte plant. So that's showing you when you're looking at these slides later that the green gooey stuff there, those green blobs, that's the gametophyte generation. Also, like liverwort, super small, even smaller than the liverwort. So there's our good friend Dime. I don't know if I need to go through this again for anyone who missed last time. This is a very small piece of metal that you may have seen at one point in your life, but yeah, so this gives you an appreciation for how small the hornworts are. So this little part here is gametophyte part, gametophyte generation. Here's a horn, sporophyte generation horn coming out. So very, very small. They have rhizoids just like everything else we've looked at so far. And they grow via those apical meristems that we talked about a little bit more last time. This is a hornwort plant under the microscope slide. Sorry, this is a microscope slide of a hornwort plant, gametophyte plant. Some species are monoecious, some species are dioecious. It doesn't really, you don't have to, know, it's not, I don't care. I just want you to know what those terms mean, like what monoecious means and what dioecious means. Depending on what species you're talking about, it'll differ. But what won't differ is that the sex organs are always going to be on the upper or dorsal surface of the green flat thallus. So for example, here, it's a cross section through a microscope. Sorry, this is a microscope slide showing you hornworts and showing you some sex organs. So again, hey, I'm going to ask you a question. So this is some sex organs here, right? Based on what you know so far, what sex organs are these? So first of all, the first thing you have to think to yourself are, what are the sex organs in plants, right? And how many are there to choose from? Well, sex organs that produce sperm, and there's sex organs that produce egg, right? So looking at these sex organs here, do you think these sex organs produce sperm, or do you think these sex organs produce egg? Okay, so is it easy? I think, is that, I don't know if we've gone through how you like to be pronounced. Okay, cool. So EC says sperm, right? So what does anyone else think? So we have the choice. Now, oh, Kiro says egg. Now Hawa says egg. Oh, why? So what? So what are, just so we, all right. You have sex organs that produces egg and sex organs that produces sperm. I'm going to draw a sex organ on the left here. Hala says sperm. I'm going to draw a sex organ over here. Yep, egg stays in it, don't move. You're right. All right, on the left here, I'm going to draw a sex organ. All right, what did I just draw on the left? What sex organ did I draw on the left? Is it a female sex organ or male sex organ? Right. EC says egg. Female, right. Kiro says female. Aya says female. Hala says female. Correct. Right? Why? Because you have this swollen part, right? The venter, you have the neck, right? And then you have the egg inside. Now I'm going to draw another sex organ. Why won't it make me make dots? Oops. All right. What's this one then? If that one is the female, what is this one on the, this one must be the male, right? Right. Noha says male. Gitika says male. Hala says male. Okay. Which one does this one look like? Ah, yes. Thank you. Yeah, so EC was right. EC was right. These are antheridia here, right? For me, the way, it's not supposed to be tricky. Remember what I told you when we first learned about these sex structures? Antheridia always look like this. It's like this uniform tissue. Right, well, okay, yeah, so it's sperm. It does make sperm. Yeah, EC, you're thinking, so EC saying that you're actually thinking about the structures that hold those sex organs in liverworts. So the umbrella octopus thing, those refer to the sex structures in liverworts, not sex structures themselves. But yes, but just so you know, so yeah, so this is how I want you guys to be like, right. This is how I want you guys to be thinking, right? When you're encountering, hold on.
Oh, and also
okay.
like the umbrella and stuff is the Archegonia 4.
Yeah.
Okay.
Yeah, exactly. So that's what I was trying to explain to EC here, is that for those, and I'll write it like, don't get confused, Archegonia 4 and Antheridio 4. These things here, Archegonia 4 and Antheridio 4, that's your octopus and umbrella respectively. Those are the structures in liverworts that have the sex organs on them. But the sex organs themselves, right? The sex organs themselves, oops, black is not a good choice on black. This, right? And this, whoops. Oh my gosh, why won't it let me draw? Let me draw. Oh no. Oh, I see. Draw. Here we go. Well, okay. The sex organs themselves don't look like that. The sex organs themselves. Oh, look, see? Look. So antheridia, the tissue looks uniform. That's spermatogenous tissue. No visible egg and no visible neck. So that's how you would know that. But that's how I want you guys to be thinking when you're encountering these kind of questions. Okay, good. My draw is back. All right. Moving on. We got to move on. So that was the hornwort gametophyte plant. Now let's talk a little bit about the sporophyte plant. The sporophyte plant is shaped like a horn. That's where they get the name hornwort. Thin horn. Sporophyte plant shaped like a horn. The sporophyte plant, this is the first time this is the case. In the case of the hornwort, the sporophyte plant is actually photosynthetic, green. We have not encountered a photosynthetic sporophyte plant yet in this class, because when we looked at the mosses, true mosses and peat mosses, only the gametophyte plant was photosynthetic. When we looked at the liverworts, only the gametophyte plant was photosynthetic. Now in the hornworts, both the gametophyte and the sporophyte generation are photosynthetic. That's an evolutionary leap. It's shaped like a horn, and so another difference in the hornworts is that the hornwort sporophyte does not have a seta. Instead, it has something that's called an intercalary meristem, which is located between the foot and the capsule, and I'll show you what that means on this slide here. So first, notice down here, right? So we've taken a hornwort and we've used it to make a microscope slide. And so the area on the hornwort that we're looking right now is in this red circle. And notice here, it says sporangium grows from the base. So the sporangium is growing in this direction. that horn-shaped thing, right, is growing this way. It's growing like that, away from the, or up from the gametophyte plant. The reason I mention that is because of this thing here, right, intercalary meristem. So if we were to look at the microscope slide, so here is the foot of the hornwort sporophyte. We just learned it does not have a seta, so there's no seta here. It's just a foot and then the capsule, which would be the horn. Whoa, let's not draw it like that. Foot and the capsule. And so, as I said before, right, the capsule or the horn is growing up in this direction. And so where the new cells, where mitosis is occurring, is actually at the base, because that's where it's growing from. So this is where the new cells are this is where mitosis is occurring and new cells are growing that horn. And that's what we call an intercalary meristem, meaning it's not a meristem up here. It's not an apical meristem, right? It's not at the apex, it's not at the tip, it's at the base. Why that's important is because, so hornwort, these are very thin, the sporangium is very thin horn. And so again, if like a deer or some other animal was like walking through the forest and they broke this sporophyte plant off, it doesn't matter because the sporophyte plant is just going to grow from the base more. So if it breaks off, it can grow more. You may think this is a whole new concept to you, this whole idea of an intercalary meristem, but have you ever mowed lawn, or have you ever seen somebody mowing the lawn? If you have, yeah, exactly, right? Yes, kind of like hair on your head, exactly. It's growing from the base, not the tips. So perfect analogy. You can cut your hair and more hair will grow, right? Same idea with mowing the lawn. If grass had the meristem at the tip, every time you mow the lawn, you would kill all the grass. So grass also has this thing called an intercalary meristem where it grows from the base. Yep, cool. Just seeing here real quick. All right, let's move on. So this was a microscope slide from the side, right, showing the sporophyte plant growing from the gametophyte plant, right, as shown in that little red circle. Now we're looking at a cross section through a horn, right, through the sporophyte plant. See, you can like this. So if you do a cross section through sporophyte plant, you notice these different layers. I'll use pink. So you have a 1 cell thick epidermis that helps protect. But the other thing is, unlike the other plants we've looked at so far, this epidermis actually has a cuticle, which is kind of a waxy thing. And that cuticle makes it so that CO2, water vapor, and oxygen cannot move across this. So if I'm CO2 and I'm trying to get inside, I won't be able to get inside because the cuticle of the epidermis is kind of waxy. Same thing for like water vapor. Like, oh, here I am trying to, oh no, blocked. I got blocked. So that's kind of a problem because you do need CO2 gas to be able, oh, and likewise, right, oxygen that's being made from photosynthesis tries to get out. Oh, nope, sorry, blocked, can't get out. That's kind of a problem because you need CO2 gas to come in to be able to do photosynthesis, and you need oxygen to be able to go out. So we have to figure out a way around this, right? Because that's not going to work if it's impermeable to these things. Anyways, I got distracted there. Here we go. Epidermis, 1 cell thick. That's here. Cortex region, that's photosynthetic. I'll do it in green since it's photosynthetic. That's this area. And then the cells inside of the sporophyte horn, right? Sporophyte plant horn that are gonna make spores, sporogenous cells. Those are located here in a ring. So this whole thing is sporogenous cells here. And then in the very middle, there's like this other thing called a columella. Oh, that's not a good color. I'll use blue, which is just some sterile region, sterile tissue. I think it probably serves as structural support to help the horn stay up. Okay, now we're gonna look, this is showing you the same, pretty much the same thing, but from a longitudinal thing. So just to get oriented before I go on to these other things, right? Epidermis would be here, right? Here's epidermis. Cortex region would be here, right? Cortex region. Central column of sporogenous cells. I think I used orange. That's all these, like, so here's a spore. Here's a spore. So this whole thing here is like the central column of spores, but then that sterile area is like in the middle. Anyways, just to get you oriented of what we're looking at here. But remember, I said on the previous slide that this having an epidermis, right, that has a waxy cuticle that prevents gas exchange is a problem. So how does Hornwort solve that problem? The way it solves that problem is with a new structure called stomatal apparatus. So I think my head, yep, my head is blocking it, but there is a stoma here, part of the stomatal apparatus. We'll learn a lot more about the stomatal apparatus, but on the subsequent slides. But just know this is another evolutionary leap in that now we have something to do gas exchange on the sporophyte plant. What else? Right, so this sporocytes, right, 2N, that's all these, this area here that will go on to make the spores. And in this slide, the spores are already present, right? So like here's a spore and you can see that little mark on it, right? Because it's a tetrad. And then just like everything else we've seen, the spores are released into the wind, except because the oldest tissue is up here, that's where it's going to be opening first. And so spores will be released from the top down into the wind. All right. Man, I got to move this chat box and I think my head is blocking this. So stomatal apparatus, what is it? Stomatal apparatus is a structure in the epidermis. It consists of 3 cells. I'll use yellow. These guard cells, which I'm drawing here. Sorry, 2 cells, and then a hole. So the guard cells, 2 guard cells, and then a hole. A stoma just means a hole, stoma. So here's the hole. Oh, it's not a good color. Stoma, right? Here is the hole. And so what the stomatal apparatus does is it regulates gas exchange. And what's cool is that, remember when we learned about the liverworts having pores and air chambers? The pore is not very efficient because the pore is like an open window, right? It's like a window with no glass. But the stomatal apparatus is like a window that you can open and close. So these guard cells that I drew before here, right? Those are gonna like be able to swell and then also get smaller. And depending on how much how swollen they are, the hole will be open or not, right? And so that helps regulate gas exchange. Again, we'll learn more about them in other classes. But that's a big evolutionary leap and a difference between the liverworts. Liverworts have pores and air chambers. Hornworts have stomatal apparatus. This is another nice drawing, shows you all the different parts. So like here, flat green thing, Gametophyte generation. And then coming out of the gametophyte is all of the horns. That's the sporophyte generation. And this slide is also showing you, like what I said on the previous slide, previous couple slides, that the horns mature from the top down because they're growing from the base. And so the mature spores are going to be released from the top. Okay. And then recall that intercalary meristem. So this is where that would be located, and the horn can grow upwards from that. All right. Hornworts don't have elaters, they have pseudoelators, which you might see in a microscope slide. We think the pseudoelators do the same thing as the elaters in that, oh, so just so you know, so like this here is a horn that has been crushed under the microscope slide, and so all of the little spores can get released. If you were to zoom in at a higher magnification, this is what you would see. You'd see all these spores, but then you'd also see these like weird things, little weird kind of like hook-like squiggly things, which we call pseudoelators. We think they serve the same function as an elater, but we're actually still not sure what they do. So we think it's like an elater. This is kind of, to sum up, this is like an alternation of generation slide for the hornworts, because I don't think your book gives you one. But it's just so you know what you're looking at. So like, here's a spore getting released, right? The spore is going to land somewhere and grow into the gametophyte plant. Gametophyte plant, depending on what species you're looking at, might be monoecious or dioecious. If it's monoecious, right, one house, there will be male sex organs and female sex organs on the same plant. Male sex organ has the sperm producing cells. Female sex organ has the egg inside. Eventually sperm via water will swim to the egg and fertilize it, right? When that happens, the new sporophyte generation will grow. In the hornworts, it's in the shape of a horn. There you can see it here, growing, right? If we were to look at the sporophyte generation under the microscope slide, you would see some of our similar players, right? A foot, that's the foot here. And a capsule, which is the horn. But we learned a new structure here, this intercalary meristem, which is where cell division happens and the hornwort can grow the sporophyte. Okay, so I'm just trying to think in the interest of time. I do think we need to start a little bit of lecture 5, because I think this is, oh yeah, there's 2 more things here. All right, because we really do need to kind of make up a little bit for lost time. So this is the hornwort, like alternation of generations in the hornwort. Oh, notice the right stomatal apparatus. So that's where that gas exchange is going to occur on the sporophyte plant as well. This is another thing. So as we go through this course, there's a lot of plants that have this symbiotic relationship. So symbiotic relationship means, symbiotic relationship is kind of like a mutualistic relationship. So symbiotic means that each player in the relationship gets something out of it. So in the case of the hornworts have a symbiotic relationship with a cyanobacteria called nostoc. Cyanobacteria is a photosynthetic bacteria. And what happens is, so the nostoc actually lives inside of the hornwort gametophyte plant. And what the nostoc does is it fixes atmospheric nitrogen. What that means is it takes nitrogen gas out of the air and turns it into a type of nitrogen that, that the hornwort plant can use for fertilizer. And like I said, the nostoc bacteria lives inside of the gametophyte plant, and the nostoc provides nitrogen to the gametophyte plant. And in return, the hornwort plant provides a nice house for the nostoc. So this on the right here, this is a close-up of nostoc bacteria. Kind of looks like green pearls. We'll see nostoc, or we'll see other symbiotic relationships in other plants too. But this is our first example of it. So nostoc lives inside of the hornwort gametophyte plant. Nostoc provides nitrogen to the hornwort, and the hornwort provides a nice house in the form of a nice house for it. All right, this is the last slide for lecture 4, where you can compare the sporophyte generations of all the non-vascular, well, except for peat mosses, but most of the non-vascular plants that we've covered so far. So in the middle, you have true moss, right? The green fluffy is the gametophyte plant. We're only going to focus on the sporophyte plants here, but green moss, sporophyte plant, green moss, true moss, sporophyte plant has a foot, has a seta, has a capsule, right? That's true moss sporophyte plant. Liverwort sporophyte plant has a foot, a very short seta, because it hangs upside down, and a capsule, right? So true moss, foot, seta, capsule. Liverwort, foot, seta, capsule. Remember, right? We're talking about the sporophyte. Talking about sporophytes. Today we learned about hornworts. Hornworts have a foot. It's not a good color there. Hornworts have a foot. No seta. Instead, they have this thing called an intercalary meristem that allows the capsule, which looks like a horn, to grow, right? This is a good way to kind of organize your thoughts, is to make comparisons and build upon what we learned already with other plants, right? Because there will be a lot of repetition. Cool. Don't get excited. We are going to start, we're going to just briefly start lecture 5 to give us a little bit of a jump, since there's still like 20 minutes left in class. And but just so you know, since this here is the end of lecture 4, so you know there's going to be an assignment that covers lectures 3 and 4, right? So 3 and 4 are the mosses. Sorry, 3 is the true mosses and peat mosses. Lecture 4 is the liverworts and hornworts, which we just finished. And there's going to be an assignment for that due next Thursday, so a week from today, before 10 a.m. So you know. Let me get out of this. And let me pull up, and we'll just take a little break for a second. If you guys have any questions, you can chime in. And yeah, okay. Yeah, I am tired, and I would like to kind of maybe not keep going, but I think it's in our best interest to keep going, so that you have, I like for you guys to have the lecture material before you have lab. I think it makes labs more productive. I've had classes in the past before where, like, I'll have lab, but we didn't even cover that stuff in lecture yet. And so then you're in the lab and you're kind of like, I don't know any of this stuff, which I always thought was really frustrating. So that's why I think we will push forward so that we'll be prepared. Okay, I'm gonna share this. All right. And we're gonna start, we're gonna start this lecture 5. We're gonna start it. We won't get too deep into it, right? We're just gonna get started. So we just finished lecture 4, which is those liverworts and those hornworts. Now, those were examples of non-vascular plants, right? So today we're gonna talk a little bit about vascular plants and start to get to know them a little bit. And what are we gonna, well, we won't cover all this today, but we'll cover some of this right now. We're gonna talk about why did we even have vascular plants show up, right? Non-vascular plants that we just looked at, mosses, liverworts, hornworts, they do fine. They've been around for 400 million years. They're out there right now. They're doing fine. Why is it important to even have vasculature? So we'll talk about why that occurred. And then probably not today, but next class we'll talk about some anatomy, and then also some examples of early vascular plants. So we finished just now those non-vascular plants. We're going to move up through the plant kingdom into the vascular plants, and the first ones we'll encounter are what are called spore plants. So these are plants that do not make seeds. There's still no seeds. They're plants that release spores, just like the plants we've just covered. And those would be ferns, lycopods, and horsetails. So the first thing we got to talk about is like, why go, right? Why go from a non-vascular system to a vascular system? What are the benefits of doing that? What are the disadvantages of doing that? Well, so first we'll talk about the non-vascular plants. If you're a non-vascular plant, you know there's no vascular tissue involved. You know that the gametophyte plants are dominant. And you know that the sporophyte plant is attached to and dependent on the gametophyte plant. You already know those things, right? So what are the advantages of that? So the advantages of that is that you're really super simple. And if you're really super simple, you don't require a lot of resources. So these plants, mosses, liverworts, and hornworts, very small. They can grow on a rock, right? They can grow on the side of a building. They're super, super simple. They don't require a lot of resources. So that's great. And that's why they still live in the world, because really simple. But there's some disadvantages to being a non-vascular plant. So number one, you're not efficient. So because you're so simple, you're very inefficient. You can't get very big. If you don't have any vascular tissue, you're limited in size. So you have to be super small. You're releasing spores just out into the wind. And so they're not protected in any way. You need to have water in order for sperm to swim to the egg. Well, that's not very efficient, right? Because what if there's no water? I guess no fertilization then, right? That's not good. And you only have one sporangium per sporophyte plant. So that's also not great, right? That's not great. Hence vascular plants. So vascular plants, you compare them, look at the, so here, look, non-vascular plants, no vascular tissue, free-living gametophytes, reduced sporophyte. For the vascular plants, this is all going to be switched. Vascular plants have vascular tissue. Vascular plants have free-living sporophytes that are dominant. Oh, totally different, right? Opposite. And vascular plants have a reduced gametophyte. So that's the opposite, right? Yeah. All right, so why be vascular? Well, you're you are more complex now. And so you could actually be really efficient and have a much larger size. Your spores are going to be protected from the elements, which we'll talk about that next time. You don't need fertilization. You don't need water anymore for fertilization. So you can have fertilization occur without water. That's nice. And on each sporophyte plant, you can have multiple sporangia. So there's a lot of pros. There's a lot of advantages to being a vascular plant. The only downside is because you are so complex, you require a lot more resources. So in case you haven't noticed, okay, there's like a fly. In case you haven't noticed, the pros and cons are reversed, right? So the pros for the non-vascular plants are the cons of the vascular plants and vice versa, just in case you didn't know that, right? Okay. Let's see here. Okay, yeah, we can do this and then we'll break for the day. All right, so that's why we have vascular plants. When vascular plants first showed up, so this is what it may have looked like. This is like a prehistoric forest. You can see it kind of looks like Jurassic Park kind of vibes, right? But one thing you might want to notice in this old prehistoric forest is look at this giant bug, right? Look at this giant bug. And that's because there's something significant about the fact that we had these early vascular plants. So here's a timeline. Let me use, I think, black. Here's a timeline. So we already covered these, right? The bryophytes, the mosses, liverworts, and hornworts. And we said that those showed up, you know, the ones we looked at, like, let's say about 400 million years ago, a little bit more even. It's like going, it's off. So you're looking maybe 475 million years ago. And then if you look at the fossil record, later we get these seedless vascular plants, or the spore-bearing vascular plants. And those showed up about 70 million years later than the bryophytes, right? So you're looking at like around there, the Paleozoic in the Devonian. So why I'm showing you that is because here we have these vascular plants that can get really big and that can live in environments that don't require water for fertilization. They can kind of spread all over the land. And what happens is as those plants spread over the land, so this is a different slide, but it's showing you the same thing, right? Like, so here's where those first vascular plants are showing up. And boom, here's on the same thing. Look, the first vascular plants show up. Then around 350 million years ago, we start to see these kind of like tree-like plants that show up. And look at what the fossil record also shows us in terms of transition. When we have these first land plants, right, a lot of the things that we see in the fossil record were just things that lived in the water, because the plants originated from the water. And when they first started to go on land, most of the animals were living in the water too. All of the animals were pretty much living in the water too. But as the plants moved on to land and became more robust and being vascular plants, so too do we see a transition of animals moving onto the land as well. So look, the first amphibians, first insects, first reptiles, and then a lot of diversification of other animal types as plants moved on to land. So what's interesting and cool about this is as the plants began to dominate the land, so too did the animals move onto land and were allowed to really diversify. I think because it's a lot of information. And I'm tired of talking to you. I think we'll pick up here next time, talk more about those first vascular plants. But at least now you can see kind of where we're going with it, and the differences in between a non-vascular plant and a vascular plant, and the importance of animal life on land as vascular plants moved all over the land. We'll pick up there next time. So if anyone has any questions, I'm happy to answer them now, or I will see you guys. So today you have lab on Monday, you have liverworts and hornworts on that day. A week from today, you have an assignment due that's on liverworts and hornworts. It should be available this afternoon. And you can do it anytime before you have a week to do it.
So there's nothing due for the lab, just kind of studying for the exam?
Ask that again.
Is there anything due for the lab, or is it like the what we learn and do there is to study?
Oh, that reminds me. I have to post. Good reminder. Yeah, so I'll post the lab material on Canvas right now. And what you should do is you should download a copy of the lab material. And before coming to lab, you should read it over. You just make a note here to post the lab 2 material. Also, Hawa saying the answers to assignment 1 aren't up yet, so I will make sure those are open. But yeah, Henry, you download the lab material, you read through the lab material, and have an understanding of what you're going to do before coming to lab, and then that's all you have to do for lab prep.
All right, cool. I hope we hit the microscopes again.
Yep, and if you have any other questions about lab, you can feel free to email Bruna or Dr. Fonseca. Her email address is on the syllabus as well. And the syllabus, if you haven't read over the syllabus yet, please read over the syllabus because it has a lot of information there about like things like that.
Thank you, bye.
Yep, thank you. Yeah, so I will post that lab material right now, and I will open up those answers for assignment 1. Everyone have a good day, have a good weekend, have a good lab on Monday. And I'll see you guys later. Thank you guys. Bye.